Hola, ¿sí me escuchan? Muy buenas tardes a todas y a todos. Les agradecemos muchísimo su presencia hoy aquí en el auditorio del Instituto de Biotecnología de la UNAM. También les agradecemos mucho a las personas que siguen esta transmisión por los canales de YouTube del Instituto de Biotecnología, de Ciencias Genómicas y del Instituto de Ecología. Es un gran honor para la universidad tener hoy aquí a la doctora Joanne Chory para dar esta charla magistral en el marco de la investidura como doctora honoris causa por la UNAM. Este es un honor que la universidad le otorga a un muy selecto grupo de académicos y personalidades de la ciencia y la cultura cada 2 o 3 años desde 1910, si no me falla la memoria. En esta ocasión contamos con, vamos a tener la investidura de los doctorados honoris causa el próximo jueves en una sesión extraordinaria y solemne del Consejo Universitario. En este año se otorga el doctorado honoris causa a 12 personalidades, de entre las cuales está la doctora Chory. Yo también quiero agradecer muy sinceramente la colaboración para tener esta charla magistral hoy, por supuesto al Instituto de Biotecnología que nos alberga hoy en su casa y al doctor Christian Solenkamp, director del Instituto de Ciencias Genómicas, por también la colaboración en la organización de este evento. Yo soy Ana Escalante, directora del Instituto de Ecología y que soy la coanfitriona de la doctora Chory en esta investidura del doctorado honoris causa. Y con eso quiero darle la palabra al doctor Solenkamp, que nos va a dar una breve introducción de la impresionante trayectoria de la doctora Chory. Muchas gracias.
Bueno, sí. Pues buenas tardes también a todas y todos por mi parte. Gracias, Ana, por las primeras palabras. Y voy a permitirme leer una pequeña semblanza sobre la doctora Chory para que conozcan un poco más de ella. Everything's nice that I'm going to say, so don't worry. La doctora Chory es genetista de plantas, es profesora y es directora del Laboratorio de Biología Vegetal, Celular y Molecular del Salk Institute en San Diego, California, e investigadora del Howard Hughes Medical Institute. Ella inició sus estudios superiores en biología en el Oberlin College en Ohio, donde se graduó con honores. Obtuvo el doctorado en microbiología en la Universidad de Illinois en Urbana-Champaign en 1984 y entre este año y 1988 fue becaria postdoctoral en Harvard Medical School en el laboratorio de Fred Ausubel. En 1988 ingresó al Instituto Salk como profesora asistente. En 1994 trabajó como profesora asociada y en 1998 fue nombrada directora de investigación. En 97 se vinculó al Howard Hughes Medical Institute y en 1999 fue nombrada profesora de la Universidad de California, San Diego. Yo personalmente conocí a la doctora Chory como profesora en un curso llamado Arabidopsis Molecular Genetics en el Cold Spring Harbor Laboratory y la admiro desde entonces, la verdad, mucha admiración, muchas gracias. Y la doctora Chory es considerada una de las biólogas de plantas más influyentes de la era moderna y una de las científicas más innovadoras de nuestro tiempo. En 30 años de trabajo ha sido pionera en el uso de la genética molecular para estudiar cómo las plantas cambian su forma y tamaño y cómo responden a diferentes entornos. Me permito mencionar aquí un breve ejemplo. Por ejemplo, para dilucidar el mecanismo por el cual las plantas responden a la luz, ella adoptó un enfoque audaz. Después de mutagenizar semillas, las dejó germinar y crecer las plántulas en la oscuridad. Esperaba que algunas plántulas portarían versiones defectuosas de genes que normalmente participan en la vía de señalización de la luz e ignorarían la oscuridad. En otras palabras, ella pedía a las plantas que se desarrollaran en la oscuridad como si estuvieran expuestas a la luz. Y como era de esperarse, la mayoría de las 100.000 plántulas tenía la apariencia larguirucha típica de las plantas que crecen en la oscuridad. No contenían cloroplastos, los organelos en los que ocurre la fotosíntesis, pero unas pocas docenas crecieron bajas y gordas y sus hojas se expandieron. Fueron más pálidas, pero en muchos otros aspectos se parecían a las plántulas crecidas en condiciones de luz. Y a partir de estas mutantes se podían identificar los genes mutados. Y con enfoques como de este tipo, ella ha logrado descubrir varias rutas de señalización y desarrollo en plantas. Bueno, continuamos con la semblanza. A lo largo de su carrera, la doctora Chory ha publicado 224 artículos de investigación, más 66 revisiones arbitradas y 15 capítulos de libros. Tiene más que 80,000 citas, un factor H de 140 y 10 patentes. Ha recibido el reconocimiento de la comunidad científica a través de diversos premios y galardones de prestigio. Junto con su pasión por la genética, la doctora Chory se esfuerza por inspirar a las mujeres jóvenes a convertirse en científicas y trabaja constantemente para mejorar el campo de acción de las mujeres. Habiendo cosechado durante décadas una gran cantidad de descubrimientos sobre cómo las plantas se modifican a sí mismas, ahora ella está aplicando lo que ha aprendido en las plantas. Ha embarcado en un proyecto que tiene como objetivo combatir el cambio climático y mejorar la productividad agrícola del país, del cual nos va a hablar hoy en su seminario. Bueno, no dice, pero dice aquí, Fighting Climate Change with Plants, It Takes a Global Village to Solve a Global Problem. Bueno, otra vez, it's a great pleasure to have you here, Joanne, here in Cuernavaca, and we're looking forward to your presentations. And afterwards, we will have some questions from here, also from the chat. So we are transmitting in YouTube channels. So we will select a few questions afterwards, and you have the opportunity to ask afterwards. So please go ahead. It's all yours.
Buenos dias, señores y señores. Muchas gracias to Anna and Christian for helping me make this trip down here. I appreciate that very much. And I'm embarrassed to say that I don't speak a word of Spanish, so I don't even know what you just said about me. But I'm assuming it's good. Anyway, we'll see. When I run it on Google Translator, it'll be interesting to see how they translate that. Okay, so here I'm coming here today with basically a message that is all hands must get on board, and about climate change. And so I think I really want to stress that point, because without everybody getting on board, it's going to be a big problem that we're not going to deal well with as the earth deteriorates. Okay, so now we have the next slide. So here's the current situation. We're at a crossroads, okay? And we have all these problems because we have 8 billion people on the planet. So this burgeoning population has moved out a lot of ecosystems. I hear some feedback. You see that? Okay, I'm sorry. All right, so, and this has increased demand for food, feed, fuel, and fiber, which are products of plants. But the products of plants are coming from these stressed out ecosystems. And that's all a big issue. And so with CO2 rising in our atmosphere, this is causing warming. And warming leads to chaos in the environment, right? And so the United Nations has said, our action or inaction in the next 10 years will determine our fate. And they named the year 2030, but it's now 2022. So it's our next 8 years. All right. So anyways, it's number 8. That's hardly any time at all. We're no longer doing this for our children and grandchildren. We're doing it for us, right? All right. So I'll have the next slide. So in the whole climate world, CO2 is kind of considered, the antagonist. It's like the bad guy in the movie. But as a plant biologist, I view CO2 as a friend. And I think, CO2 is a friend because it basically is food for plants. And we're a carbon-based society. Can I have the next slide? We're a carbon-based society, so plants become the major drivers of our whole natural carbon cycle. And then that leads to all the biomass that we have on Earth, which is almost all carbon-based, right? All right, so why did we come up with plants as our idea? we think nature is the best solution because plants have evolved over, these are land plants, not just, photosynthesis evolved 4 billion years ago. And the land plants came out of the oceans and lakes and stuff, and they evolved about 500, began evolution about 500 million years ago into their lineages. And there's 450,000 species of flowering plants today. And do photosynthesis, they fix carbon into sugars and different other big molecules. And we did that by improving nature's ability to store more carbon in soil for longer, we can mitigate climate change, okay? And I think that this kind of shows us on the left-hand side. All right, so here you can see plants suck up CO2. They're really good at that. And then they fix it into these molecules, but we need them to redistribute those molecules, maybe make less cellulose, which is always turning over. And when plants die and decompose, just like animals, they release all the CO2 back up into the atmosphere. So let's look at the numbers for this. Okay, next slide. So every year, 746 gigatons, 746 billion tons of CO2 is captured by nature through the process of photosynthesis, mostly through the process of photosynthesis. Okay, so, but then nature can handle most of that, and it actually fixes it back into biomass, and it does that, and then that's okay. But when the plants die at the end of the year, a lot of plants are annuals, as you know, or there's a forest fire, then the plants release all this carbon dioxide back into the atmosphere, and that number right now is at 727 gigatons. And that's released by nature. In addition to that is the 37 gigatons from humans. All right, so, and nature knows how to deal with 20 of those, but there's about 18 gigatons excess every year. And that is causing, okay, and that is causing, I need to go to the next slide. Sorry. That is causing this rise in CO2. Particles per million in the atmosphere. So this is a Keeling curve. And Keeling was an interesting guy. He measured 3 times a day the level of CO2 in the atmosphere. And he did this since the 1950s. And then they was extrapolated back to other measurements. And it's pretty clear when you look at what happened on Earth, human activity as a result of the Industrial Revolution caused this thing to happen, okay? So if you went back prior to 1960, the curve is almost flat, all the way up, way past 1850, when it normally started as the start of the revolution, the Industrial Revolution. But you can see in 1960 there was a turn, and now we have this big slope, and the models don't accommodate this at all. So everyone thinks it may be worse than we actually think. Maybe 8 years is really, we can never return. But I still have hope. And I want to convey that to you today. But I think hope means for me that I got to work on this problem. Okay, so next slide. All right, so we sat around and talked, and we came up with some ideas of how we can use plants to help us get rid of some of the CO2 in the atmosphere, right? Okay, so we're making now what we're calling Salk ideal plants. All right, so on the left-hand side, am I shaking the table? Okay. On the left-hand side, you can see what the normal plant does. It sucks up CO2, and then it dies and decomposes, and it releases it back up. But our plants are designed to keep more CO2 in the plant, and in the plant root specifically. Okay, and we're going to keep it there in the form of a recalcitrant polymer that all plants make, called suberin. So in my talk, if you have a take-home lesson, it's just that we're going to work on 3 traits. We're going to alter 3 traits, which we hope is going to alter the amount of organic carbon that we can bury in the soil, which is a great sink for burying carbon that is the result of CO2 fixation from photosynthesis. Okay, so here's the 3 traits. So we want to, this is the center of the show. We want to make more recalcitrant carbon-rich polymers like suberin, and I'll show you what it looks like in a second. And this will enable our ideal plants to store carbon deeper in the soil for longer if we add these next 2 traits. So we need to make plants that make more suberin directly. We also want to make more suberin indirectly. Okay, and by that I mean we want to have more massive plant roots. So if we make more massive plant roots, there'll be more carbon in those roots, and they should have more suberin. That was one thing we thought, and that's right. That does work out. And then we have deeper plant roots, and this is something we want the plants to do. We want them to burrow down their root deep, because if you get below 3 meters, I think that's the right number, or 60 centimeters. I can't remember. All right, I think it's 60 centimeters. I take that back. Sorry. So if you get down below 60 centimeters, soil becomes a little bit anaerobic. Then it gets even more anaerobic, and there aren't many microbes that live down there. From the few samples that have been taken at that depth, there haven't been many samples taken, but the soil is itself going to be a really good place down there to store a lot of carbon into it, right? Because you need a place where the microbes aren't going to release it all. Okay. So here's the structure of suberin shown on the right-hand side. And as you can see, I mean, suberin is a pretty complex structure. And it's the middle part that we became most interested in, because everywhere you see a black dot there, those are carbons. So this is a good carbon storage device, right? And everywhere where you see a red dot, those are oxygens. And there aren't many of those. And that's the entry point for microbes to come in and break down big molecules made by plants. Okay, so, all right, so our idea is the suberin is going to put us in a good place to start. If we can get the plant to make more suberin and efficiently store it in the soil, as if we have to get farmers to quit tilling too, that's part of this whole process. But I think that's happening already. So this shouldn't die and decompose, and it should efficiently. It doesn't decompose quickly, and efficiently stores carbon in the soil. That's what we want to make suberin do. All right, next slide. Okay, so I can tell you the first experiment we did, we thought it was a good one, but it was a bad one, okay? Because what it told us was that, well, others know where suberin is made in plant roots. And so here on this left-hand side, you can see a wild-type plant. And this is an Arabidopsis seedling, I think, maybe a little older than a seedling. And you can see there are 2 concentric circles. If you do a cross section, do that root. Okay, so and they're stained red because there's a dye that stains suberin red. So you can see here that suberin is expressed only in certain tissues during development. So what we did was, there was a transcription factor been reported in the literature, and this transcription factor was thought to regulate the whole suberin biosynthetic pathway. And this is something that came out from other people's work on different pathways, but it seems to be a pretty good rule of thumb. If you want to make more of a plant product, find the transcription factor that controls the whole pathway. So plants have put these pathways under the control of like a major regulator. And so we tried that transcription factor with the 35S promoter, which I'm sure some of you know what it is and some of you don't, but it's mostly ubiquitously expressed in the plant in every cell type. And when we did that, the plants starved themselves to death. Okay, so that's not a plant that's going to help us carbon sequester, right? So we do that one by the wayside. And then we said we better learn more about roots. So now we have identified a lot of promoters that are in these cell types of the root, and I'll tell you what cell types they are in 1 second. And we're starting to look at how those plants vary in their suberin content. Okay, so I'll take the next slide. So here are the 2 cell types that are making suberin. Okay, so in this case, This is the primary growth of the Arabidopsis root. And no one ever studies secondary growth of Arabidopsis root. I learned that. But anyway, that's going to be important for our studies. So you can see here where the orange line, no, I think it's the orange line. Hang on. Yeah. All right. It's the yellow ones. The yellow cells are the endodermis. And the endodermis is responsible for making suberin. And when you make suberin in the endodermis, you can block out a microbe, or you can block out water, or you can keep water in the plant. So suberin is like a sealant. It really does a good job at that, and it really affects water relations, which is an important thing to know about it. Okay, and then on the right-hand side, you can see when the plants get older, they make a secondary growth, and then that growth is called the periderm, and that's shown along the top. So the periderm is secondary growth that has its own root meristem. And then, so this now is growing from the pericycle, and when the plants get older, this becomes the major place where things get blocked. And that keeps stuff in mostly. All right, so next slide. So what we did was a GWAS, and this is genome-wide association study for the onset of periderm development. Our idea was if we can make more periderm, then maybe we could make more suberin in the plant without killing it. And that turns out to be right, okay? So here's 5 chromosomes of Arabidopsis along the bottom, and you can see that some of these dots are pretty high up over this line at the bottom. And so those are telling us those are significant points where this GWAS finds an association. And this study was done by Gonzalo Villarino and Signa Dahlberg Wright in the Bush Lab. And what they used were the 1,130 strains of Arabidopsis, which are available with soil samples and GPS markers, so you can go back and find those. It's important to be able to find those strains so that you can get them back out. All right, so Gonzalo cloned this gene because we can do that kind of stuff easily in LM. All right, that's the same slide. Okay, next slide. And it's a transcription factor. Okay, and the phenotype of these plants is shown on the next slide. This is the transcription factor in green. All right. Here's the next slide. And here's what the phenotype looks like. So on the left is the wild type, or a wild type Arabidopsis, one of those accessions. Okay, and so you can see here, the wild type has this one layer of periderm. And on the right, you can see the plant we engineered based on what the hit was from our GWAS study. And the hit was for this transcription factor. And when we overexpress that transcription factor in the periderm, okay, so we had to find transcription factors which were expressed specifically in the periderm. When we do that, we get a double layer periderm. This is pretty surprising to us, but it's pretty exciting, right? Because this will definitely be a way to make more suberin in the plant. You can see that where it says second, there are 2 rows of periderm now side by side. And the way we did that screen, it was all done using automated microscopes, which worked day and night for us, right? And we went through all of our mutagenized collections, and before that, it went through all the accessions. And so that's how we found that gene. That's one of our first new genes. Okay. Next slide. Okay, so this is just showing you how this process works. Here's plants, and you can just, okay. And we're looking at developmental time, and so it should be, the first sample is taken at 8 hours. Or 8 days, sorry, I take that back. There's no marker there. All right, so this is 8 days, the sample on the left, and then you can see at 12 days, it's not coming up, okay. All right, sorry, just click it again. Let me see what's happening. All right, that's right, leave it there. All right, so, all right, let's just look at, you're not gonna be able to see 12 and up to 24, sorry about that. Okay, so 24 is over here on the right-hand side. Okay, so now all the staining is in, that's 24 days, and all the staining is in the periderm. And in the 8-day sample, there's no staining, and at 12 days, that you can't see because the animated on top of it, unfortunately. Anyways, at 12 days, the staining is all in the endodermis, okay? So now we know sort of the developmental time course where suberin gets made, and this tells us when to do this kind of study. And in the meantime, we've identified a lot of promoters that are specific for those cell types, and I'll show you how we did that. If you just animate that, no, one back. We did that using single cell RNA-seq. And here again is the same time course. And if you look at 24 days, I think this sample is where you see red are the clusters of cells that are making suberin, that are transcribing suberin biosynthetic genes. So we think those are the cells that make suberin. Okay, and so now we have a handle on how to look at this. So a lot of what we've been doing over the last 3 years is really just preparation, right? So, okay, for the big experiments which are ongoing now. All right, so next slide. Okay. So there's another way you can make more suberin, and that was what we tried to do in that first experiment. When we took the transcription factor that controlled suberin biosynthetic pathway and overexpressed it all over the plant. But now we're going to try to do this, make more suberin per cell. Okay, and hopefully we're going to get there using periderm specific promoters. Okay. All right, next slide. So here, this shows you the suberin biosynthetic pathway, okay? And so it's a lot of it is known. It involves different compartments in the cell, and it has a big overlap with the lignin pathway. So this makes this a little difficult to figure out. But the fatty acids are synthesized in the chloroplast, and then they're like made longer and longer. So the longer you get in that middle part of the protein, that crystalline part of the protein, then the more hydrophobic you get and the more recalcitrant to break down you are. All right, so we have these enzymes which are elongating the fatty acids, and then these products meet up with some other products over here made from these pathways on the right-hand side, and the phenylpropanoid pathway is involved. And so that's a pathway we've manipulated in my lab in the past. So we know how to manipulate that pathway. And so the idea is we need to find the rate limiting step here. And so one way to find the rate limiting step is to look for bottlenecks. So if you have the right mutant backgrounds and then you overexpress a gene, you might be able to see the rate limiting step. And we've used that successfully with the brassinosteroid pathway and the phenylpropanoid pathway by doing a technique called activation tagging, which was developed by Detlef Weigel in my labs 20 years ago. Okay, so what we did was activate the expression of genes in certain cell types and then look for what the target genes were. And so we're actively trying to figure this out now. What's the rate limiting step? Because that would be another way to get more. And we also have recently started analyzing plants which are overexpressing that MIB transcription factor that was reported in the literature to have an effect on leaf suberin levels. And those plants do have more root suberin. So that's going in the right direction too. But there are also other bottlenecks you might think this pathway could have. Okay, so one of them is moving suberin across the cell membrane so that it aligns itself outside of the cell by the cell wall. Okay, so that's going to have those. That's done by ABC transporters. Again, isolated by someone in my lab, Sibel Goldstein, when she was looking for steroid transporters. But anyway, it's funny. You stay in the world of biology long enough, you get involved in every project. So I don't know. Okay. All right. And in the last place where suberin might have a problem, and it might have a bottleneck, is assembly of the polymer from the monomers that have been brought through the membranes. All right, so we're looking for all those kinds of rate limiting steps because the idea is if we get plants to make a lot of suberin, if they're in the right cell types, it shouldn't really kill the plant because a few layers of suberin should block it. But if we have 20 layers, we'll have more suberin, but it won't kill the plants. I think so. That's what we're trying to do with suberin. Okay, so now I'm going to move on to trait 2. All right, can I have the next slide? Oh, I forgot to tell you this. And here's the reason why we're so excited about suberin. And this is work done by Klaus Schmidt-Rohr at Brandeis University. And he was looking at organic soil carbon that is in peat. And peat is known to be a rich source of this stuff. And here's his solid state NMR structures for the fats relaxing components in peat humans. All right, so, and you can look at it up above, and that's the suberin from cork plants. So suberin is cork, right? So if you take the cork off the bark tree of the cork plant, off the cork tree, the Portuguese cork tree, this is what the structure of it looks like. It looks very similar. And what Joe Noel is most excited about, he's our biochemist on this project, he's most excited about these double peaks that are over here by the number 40. Okay, so you can see the first peak is actually a crystalline form of this compound, whatever it is, if it's suberin or human or whatever. So anyways, I think this is going to be really important. So the other thing we've been setting up through all this is to develop high throughput assays of suberin content, right? Because we have a lot of stuff to look at. And let me tell you why after I finish this update for you. So what I'm giving you is an update of what we've done during the pandemic in the last year. Okay, so now I'm going to tell you about trait 2. We're working on all 3 traits simultaneously. So trait 2 is more massive plant roots, right? So how can we make the root a better sink, or the young leaves where photosynthesis is happening, a better source of carbon that goes to the root? Because if you're going to make more root, you have to have more carbon down there to make it from, right? And so we have this idea based on old literature that cytokinin was involved. So nowadays, the rate limiting step in cytokinin biosynthesis is known. So we overexpress that biosynthetic gene in a certain cell type of the leaf. And we think what we did was create a young leaf that was a better source of carbon that was getting out of the leaf into these sink tissues, which are the root and the inflorescence. Okay, so that looks like another good hit for genes that might be involved in carbon sequestration. All right, next slide. Okay, so here shows the plants that we made when we did that. These are Arabidopsis plants. They're genetically identical except for one gene, and that gene is the cytokinin biosynthesis gene. And you can see here the plant on the right, which is overexpressing that gene, is making a lot more root, right, than the plant on the left. Okay, so we like this one. This is staying in our pipeline, and we applied for patents for it and stuff. So anyways, we'll see how that all plays itself out. All right, next slide. Okay, here's another genetic change a single genetic change that resulted in increased root biomass. And here you can see how much root is being made on the left versus the other 2 plants. And that's pretty impressive, I think. Okay, and the other 2 plants are Arabidopsis not overexpressing this microRNA. So this is a microRNA now. So again, it's a different pathway, I think. All right, so, and I think one of these are being overexpressed in just the root, but I'm not sure of that, so don't hold me to that. Okay, so again, when we overexpress this gene everywhere in the plant, it does does some things bad to the plant. Okay, so you get more root, and there's a lot more root, and the roots are longer. So those are all good traits for our carbon sequestration screens, but we have to feed the world on the plants that we're going to make carbon sequestration plants. And I'll tell you what those are in just 2 minutes, I swear. Okay, so this is a little bit worrisome about this plant, because if you look at those seed pods, I don't know how well you can see it. Those seed pods on the plant on the left are barely in existence. Normally a plant that made that much shoot, that's a single plant we're looking at there. Normally a plant to make that much shoot would have a lot more seeds, right? So the yield has gone way down on a plant like this. So this was bad. So we had to go back and look at root specific changes, and we think we have some good promoters for this now. And just I want to tell you about these examples because you have a good idea, oh, you do a meet and screen, but you can't just assume it's gonna work, right? So anyway, okay. All right, so here's the second pathway that increases root biomass, and I'll show you two more. I'll show you them fast. All right, next one. Okay, so here's more root biomass changes. And here we're using Arabidopsis as a weighted assay for beneficial endophytes. And so this is work done by Paula Saldana Guzman, who's Mexican, by the way. Anyways, and she was looking for this is not hers. Oh, she's helping out with this. All right. This is a different experiment. She's looking at endophytes, but she's helping out with this. So her name is down there. Sorry about that. And so, all right, this is endophytes. All right. I'm sorry, I'm getting confused. But I'm back on track now. Here we go. All right, so she put this endophyte. She purified about 100 internal to the plant microbes, okay, and then put them all back on Arabidopsis. And she asked if she could promote growth. And the answer was yes. And you can see those 3 plants on the right have received an inoculation of endophyte. And it's a pretty low inoculation too. So she puts it in the soil and they must get up into the shoot and the root because we can see the endophyte in sterile culturally grown plants, in both the shoot and the root. So anyways, it's causing the shoot and the root to be bigger. So in a way, it's a new trait, right? It's the shoot root is bigger trait. And so we have both the shoot and the root, which means these guys are fixing more CO2 and doing better photosynthesis probably. We haven't checked that yet. Okay. All right, next slide. So now we're interested in putting all these genes into crop plants. So I had Paula inoculate her plants, these other plants that we're using in the lab, and I'll tell you about them one more minute. And then here's rice on the left. And you can see here, when you add the bacteria, you get more growth of shoot and root. And then here's root in the lower right-hand corner. And so you can see there's a lot more root. There's a fair amount more root, and there's more shoots. So that's another good candidate. That makes this endophyte a good candidate for trying to do this experiment in a field trial. Okay, and then the last one is canola. You can see canola has a really big effect in the shoot. There's a small effect in the root too. The root gets a little bigger, but the shoot has a big effect. And so we need to follow that up with more experiments. Okay, next slide. Last one of trait 2. Here we're looking at a gene involved in carbon metabolism, and these made the biggest plants of all. So we have both big shoot and big root. We overexpress this gene, and we're following up on this one too. This is kind of exciting. All right, so go next slide. And now we're going to trait 3, which is find genes for deeper roots. And then we went back to these Arabidopsis 1130 strains that have been completely sequenced. And this was done by Takahiro Ogura. This paper was published in Cell about 3 years ago, before the pandemic. Anyway, here you're looking at all these different accessions of Arabidopsis, and where you see red is the root stain. Okay, so if you cut through a pot this way and look and see how deep the roots go, you can see some of these strains are shallow, and some of these strains are deeper, like MIB 60 is deep, COC 1 would be deep, Chi 0 is deep, LP 2-2 I would call shallow, I call shallow. So you can call these all something, right? And so he did a GWAS and associated genes to deeper roots. Okay. All right. Okay, so here's his gene that he found, and he edited this gene in 2 ways. He edited it to be a null at the locus, and he also edited it to have more of this protein that is made by this gene. And what he does whenever he edits it, either puts more or less of this gene product in the plant, he sees the same result. He gets deep roots. So this thing must have to be in balance with other things in the plant. So we looked at what was up with this. This is kind of interesting to us. And so what's up with this is that this plant doesn't know which way is up or down. All right, so it's probably a mutant in the auxin auxin signaling pathway, but this plant, it just doesn't know which way gravitropism is going, right? So it sends roots down deep rather than meander them around. All right, so, but that's pretty cool. So we can change shallow roots to deep roots with that gene. Okay, I'll have the next slide. And so we've also done, I'll tell you that gene is involved in auxin signaling, right? So now we have a cytokinin gene and auxin genes. And I'm going to tell you about when we did chemical genetics, we got ethylene signaling components out. That's the 3rd plant hormone pathway we've hit now. And a lot is known about these pathways, so you can think of a lot of ways to make targets that could affect carbon sequestration. Okay, so here's the chemical genetics screens. Somebody went wrong here. And you can see here, these are different chemicals that he put on these plants. That she put on these plants, sorry. And yeah, and so she was looking for plants that would stimulate and make bigger roots as a result of applying these compounds. We have a nice library at the Salk, and we have good robots. So she was able to do all this pretty quickly. All right, so here we go. All right. So what she showed, and I guess I didn't put the slides, is that the root systems architecture of Arabidopsis changed when she applied one of these compounds, and it's called S2-1. And so again, she went straight into the crop plant that we're interested in making the carbon sequestering plants. And in her case, it was soybean. And you can see she can change the root system architecture if soybean by adding this compound. Now, I don't think we're going to try to make tons of this compound and spray it on fields or anything. But what we'll do is use the compound to get at the mechanism by which it works. And I think the next slide shows one of the things it does. Okay. All right. Okay, so S2-1 is the compound she likes. And here she applies it to Arabidopsis and she can change the lateral root angle. All right, so she does that. And I think she does it in soybean. Okay. All right, the next slide. Okay, and it acts a lot like treating a plant with ethylene. All right, so that's important to know. So ACC is what you add when you want to add ethylene. You don't have a gas setup, right? And so here you can see that both of them are behaving like they're changing the root system architecture because they're changing the angle that lateral root comes out at. Okay, next slide. All right, so here's a summary of our progress. We spent a lot of time doing this stuff. So far, we have 23 single genes that affect one of these three traits that we proposed with carbon sequestration traits. And 7 with deeper roots, 6 with more roots, and 10 with more suberin. We've observed about a twofold increase for each trait by each mechanism. So by twofold increase, I mean twofold increase in suberin. Okay, so if we can combine these, this would make us have a lot more suberin. That's what we're doing right now. Of course, that's genetics, so that takes a while, even in Arabidopsis. Okay, so we're beginning engineering traits into crop plants. Now, this is something new for our lab. We've had a plant lab at Salk, since 1983. I think Chris Lamb came there, and in 1988 I joined with Arabidopsis, and we've never worked on anything else but Arabidopsis there, right? So now we have to learn how to transform elite varieties of crops. Good luck with that. We're having a hard time with that. But I have one guy with really good green thumb, and so he has gotten a bunch of transgenics out of canola and rice and stuff. And so that is looking good. Okay. All right. So we can do that. If you work hard, you can do almost anything. That's what I say. Anyway, so now we're combining these enhanced traits pairwise and by higher order magnitude. This will take a while in Arabidopsis so we can look and see the effect on suberin. And we've begun putting these traits into crop plants, and I'll tell you why in a second. Okay, next slide. All right, so I hope I convince you that Arabidopsis is a suitable model to study natural variation of root growth. I mean, we've had very fruitful screens, and I just want to make a plug for that GWAS collection that was put together by Magnus Nordberg and some of the collaborators, because that is a very powerful collection. It's really powerful for us because we can go back and look at the soil, right? Because they have soil samples from every one of those accessions. And they have the GPS site, so you can go back to the right spot. So Arabidopsis has been our premier model. It's the workhorse of plant mechanistic studies and plant molecular genetics. And so you can see these roots, how differently they grow. It's pretty amazing. I'm just looking at that one slide. Okay, so we have the second plant that we want to use, but we haven't done much with it yet, and it's Lotus japonicus. And this is because we're pretty interested in legumes, because legumes are a source of good food, and you don't need to eat meat if you eat good legumes, right? So we want to get involved with that, and we think suberin will play a role there too. Okay, so next slide. All right, so our process to create Salk ideal plants is first, we started out by finding genes, right? And so we think we need to get more genes, unfortunately. And 23 sounds like a lot, but it isn't really enough because you have to be able to go with whatever the plant is that you're growing. And what is its choice, of how it sequesters carbon? Like maybe some like the cytokinin pathway, but maybe some plants like other pathways. And so we think we need to have choices as we go in these other plants. All right, so we find the genes and we test the enhanced genes in model and target crops, right? So we have our model stuff pretty much done as we go. We have a good setup for that. And then we're transferring these stacked traits into crop plants. And that's going to be where the challenges are because we have never even done a process with any of these plants. All right, next slide. So here are the plants we chose. We chose to work with the 6 most prevalent crops there is on Earth, right? Because when we did the back of the envelope calculation, it was clear this is going to take millions of hectares of carbon sequestering crops. All right, so that's a lot of hectares, right? So the top 6 crops account for 50% of all the arable land on Earth. So we figure we can work with 6 crops. We can't work with 600 crops, so At least for now, we're sticking to these 6. And these 6 include the major calorie crops, which are wheat, which is the most prevalent crop, corn and rice. And then we have soybeans, sorghum, and canola. And sorghum is the kind of the odd man out. It's pretty much grown still in a lot of places, though it seems to be being cut back. It's a little worrisome. But sorghum grows on really marginal lands. So if you include marginal lands with the arable land, then you go up to like 6 billion hectares. That gives you a lot more space, and it doesn't interfere with food, right? Because that's the other thing that we're sitting here thinking. We can't go in and destroy yield, right? We need to go there and we need to keep yield at least at, flat, because we're asking to feed up to 10 billion people before this goes over the hump, right? So we have to take the whole food issue as part of this problem. All right, next slide. So those are our 6 crops. And here's our model, which gives us the potential impact. Okay, so if we planted, all right, let me show you an example. Circle one more. Okay, there's no circles here. All right, sorry. Okay, so look between those 2 white lines. There it is, 24%. All right, so if we want to do this globally and at the gigaton level, which is what you have to do in order to be at the right scale, then we have to get a lot of this land that we're currently using for non-carbon sequestering crops for carbon sequestering crops. So the idea is we add back these 3 traits to these crops, right? And these crops are grown everywhere on Earth because Borlaug figured out how to do that when he was in Mexico, right? And so, anyway, so they're good crops to choose, and every country has an infrastructure for helping their farmers out. And the infrastructure is all in place for seed dispersal and stuff. So this is why we pick crops. So the potential in reduction in global annual excess of CO2 is what we calculated here. So we have 18 gigatons excess, right? So if we want to get 25% of that taken out by our methods, how much land is that going to take? So we would have to do, here's 24%. So it would have to be 40% of all those crops, all 6 of them. I think this is an average of all 6. And from that, you have to, so the percentage of each plant, which is carbon sequestering. Okay, so this tells you how much more suberin you made. So if you make 2x more suberin, how many acres do you need, etc. So in this case, we're making 40% more suberin. That's a fair amount, so we have to get pretty good at it. And we also need to have 40% of all the acreage of, say, rice into a carbon sequestering crop. And that's going to give us 4 to 8 gigatons of carbon that we pull down from the atmosphere. Now, it's not clear how much carbon we have to pull down from the atmosphere. There's 18 gigatons excess per year, but there's all that carbon that's already all up there, right? We have to go carbon negative. We can't just go carbon zero. Carbon zero is not going to work because, it's just not going to work because we keep putting stuff up there. So everybody can help out by, dropping their emissions, but you still need these kinds of technology, like drawing down with plants, in order to get the carbon emissions to go negative. All right, so that's that. Now the next slide. Okay. And then there's a side benefit of all this, right? If you make plants that have bigger roots and they have deeper roots, which is they can get to sources of water that the other guys can't get to, and they make more suberin, these plants will be more resilient, right? And so additional carbon, and they'll put more carbon in the soil. When you have more carbon in the soil, you get more available nitrogen, phosphate, and some of the other nutrients that plants need, right? So this is important because we want to do sustainable agriculture, right? And we're not doing it right now. Every place we grow a lot of elite crops is completely carbon, soluble carbon, almost dead, I mean, you look at Yuma, Arizona, we have a site there. There's almost no carbon left in that soil. And they used to grow food crops there, now they're growing hay. So that's what happens, right? It becomes this marginal land, and then you can't feed people anymore. Anyway, so scientists expect the world's population to grow to 10 billion by 2050. It's coming up, which will require trillions more calories. Some estimates predict a need to double the current yield. If we have to do that, we have to do it in carbon sequestering plants, I think. So sockeye-dew plants have secondary benefits for food security. They have additional suberin and bigger, deeper roots, and that is correlated with increased soil carbon, which makes soil healthier, improving yield opportunity, reducing nutrient input requirements, improving microbial life, and enhancing drought and flood resistance. So, all right, the carbon added to farmers' soils from sockeye-dew plants will make soil healthier, enabling the soil to produce more food. And we have a commitment that our crop plants will be yield neutral or better. All right, so are we crazy? I think we are a little bit. But you have to kind of believe you're going to be able to do this, because if you don't, you just basically have given up on the world, right? I always get very emotional about this. All right, next slide. Okay, so we have field trials which began last year, and we started out in Central Valley, California. That's a source of rich soil. And Yuma, Arizona, which I just told you is a source of poor soil carbon. And then we have 2 other sites in the Midwest. We have Purdue, Indiana, and we have University of Kentucky. And these fields have historical data on them, so they're going to be very important to our measurements about soil carbon. Because it's not clear how to measure soil carbon over years to show you've actually pulled down more and stored it in the soil. Okay, so that's something we have to work on. Okay, next slide. All right, we have multiple pathways and shots on goal to achieve success. I'm an American, after all, everything's always a sports analogy, right? So we have to have shots on goal. All right, we have to have full court press, and we have to have, all those other things. Anyways, so what we have to do to achieve success is get beyond traditional breeding into more modern breeding, which is being done by several groups that are doing a great job. So what used to take hundreds of thousands of years now takes 3 years, depending on the crop. You can domesticate a crop in 3 years now. That's amazing to me. And so, and GMOs are going to have to play a part in it. I'm sorry to say that, but I think they're going to have to. Okay, next slide. So Salk solutions are credible, they're scalable, and they're sustainable. And that's why we like them a lot. So, and they're cost effective, right? So that describes some of this here. Because people really worry, and rightly so, that you're going to have, unanticipated consequences when you start messing around with the whole world, right? And I think that's a legitimate worry. So we're trying to think of every way we can create some circumstance where this might turn into something bad. So far we don't have any. I mean, we think, well, maybe we can overcool the earth, and that would be bad. But I think overcooling the earth is something we can stop with plants, because we just don't plant them anymore. So, all right, so let me finish up. All right, so here's our timeline to scale for crops. That's the name of this program, which is CO2 reduction on a planetary scale. So we're in phase 1 now, but we're also in phase 2, and we're going to begin phase 3, hopefully sometime soon. So phase 1 is the science development, research, and the research that gets done in the labs at Salk. And those are those traits I told you about. Phase 2, phase 3 is the scale up worldwide distribution, okay? So we need hundreds of millions of these hectares. We have to get farmers to plant them. We need to make arrangements with big pharma. With big ag. We need to get Bayer on our side and say they're going to plant all their corn with carbon sequestration traits. So far, they seem interested, but they're waiting for us to show them it's going to work. Okay, so we need to prove that we can increase the amount of land that we can start planting and get enough seeds so that we can plant. And so we think a proof of principle is 1 megaton additional carbon sequestered. That's 1 million tons, not 1 billion tons, right? And we think we can do that in 5 years from this year. So I think our donors are going to hold us to that number, so we really have our work cut out for us. And then finally, we think that 10 years from the beginning of field trials, which was last year, should get us to the gigaton level of carbon sequestered from the atmosphere. And phase 2 is what's going to bring that to be, right? And so we have to do phase 2. Okay, next slide. We think we have to do phase 2 in a company and not at the Salk Institute because we're not equipped at the Salk Institute. I don't know how well equipped you guys are for this kind of stuff, but we're not equipped to bulk up that kind of seed, right? And so we need to get people involved. And Sequester is a little company that we just launched. And it's going to put the world in carbon balance. We'll see if that happens. But anyways, the idea is Sequester will be the arm out of the Salk and into the ag industry. All right, so we'll see how that works. But we don't see any other way to do it. And our donors felt strongly that this was the only way to do it. They say nonprofits cannot do scale. That's what we heard that so many times, So these are things that you learn when you have to do something. Okay, so now I'll have the next slide. All right, let's skip this. Go to the next slide. Okay, so here's what I told you today. Climate change poses an immediate and existential threat to our future. We have no time to wait. The United Nations predicts that by 2030, rising global temperatures will be beyond our control. That's scary. Plants are a fundamental driver of our carbon cycle. The Earth's plants and wetlands have the potential to mitigate climate change. By understanding just a few genetic pathways in plants, in our case 3, we hope to turbocharge a plant's natural carbon sequestration process, enabling plants to convert the CO2 into a stable form of carbon and bury it in the ground for a long time. We are working with 3 traits to enable this, and our science is applicable to any plant, food, or cover crop. By leveraging the existing global agricultural infrastructure, we can scale our technology to millions of acres of arable land all over the world, achieving multi-gigaton CO2 removal per year. Okay. All right, next slide. So why did this happen at Salk? People always say Salk is a biomedical institute. The Department of Energy called us up and they said, "What are you guys working on?" And we said, we're just 5 plant geneticists, and we were trying to work on climate change using plants. And so they liked our idea, but we'll see what happens with that. But here's why this happened at Salk. It's because Jonas Salk made a place that was an inspiration to the scientists that worked there. Okay. Here's some of his quotes. So our greatest responsibility is to be good ancestors. And here's another one. Hope lies in dreams and imagination and in the courage of those who dare to make those dreams a reality. That's the quote that really has the biggest effect on me. It's where I walk over every day when I come to work. So next slide. All right, so we had a challenge by Nobel Prize winner Elizabeth Blackburn, who was our president about 5 years ago. And she came to Salk and she said, "You guys got to work on something that's bigger than just your lab, So the plant scientists took that pretty seriously. In fact, we took it the most seriously of any group at Salk. maybe because we were the most desperate, we had the least funding, you know. And so, anyways, we took that seriously. So she really presented the challenge to us and helped us think through the early phases of this project. Next slide. All right, here's the core leadership team. And we're 6 plant scientists who each have a lab at SONC. And you may know some of them. Joe Ecker is, you know, been around forever like I have. And so he's in our initiative. Joe Noel is in our initiative. He's our biochemist. Joe Ecker knows how to go to scale. And it's me, I do a lot of environmental signaling. Wolfgang Bush is our root expert. And Todd Michael is our genomics expert. Okay, so next slide. Here's his current team. You can see it's grown a lot. So that's another thing. I had to learn this year how to hire people. And it was really hard. And with the pandemic, it made it even harder. Okay, and they've all been working hard at it. And you can see many of them are pretty young. So young people feel the pressure coming from a bad climate, I think. Okay, next slide. All right, so we had to have funding to do all this. And these are the places that are funding us. So TED Audacious program kicked us off. And they kicked us off with a really big grant. And we're very grateful to them because they also provided some credibility to the project. And then from that, we got money from Bezos' Earth Fund. You probably heard of him, right? All right, so we got a grant in his first round, and it was a big grant. Then John Hess is Hess Oil. And so oil and gas are very interested in our project because they know they're going to start getting fines sometime soon. And then we've also been supported by a couple of members of our board, Salk Institute Funds, Semper Energy is our local energy company, and HHMI, who supports me, and Joe Ecker, and they pay our salaries and some salaries of people on this project. Okay, so next slide. So here's our vision. Our vision is a world in carbon balance. And I like to think about it this way. You know, in California, we have a lot of desert. So there's all these fields where they planted kind of, you know, windmills. And this will definitely reduce emissions because making electricity usually costs a lot of kilocalories of energy. All right, and on the left-hand side is showing the sorghum fields. And I like to think this is a sorghum field in West Texas, which is a total wasteland, right? And we want to plant it with sorghum that sequesters carbon and makes the carbon emissions go to net negative. And so, and we're hoping that will give us a world in carbon balance. Okay, and I think that's it. Oh, here's an easy way to remember what we're doing. 10, 10, 10. So what we want to do, we want to draw down up to 10 gigatons of CO2 per year and at a cost of less than $10 per ton CO2 within 10 years of completion of major field trials, which is 2031. So we have our hands full, but I think that's all I have to say. So thank you very much for coming. And I look forward to your questions.
Thank you so much for great talk, great inspiration. I'm sure there will be some questions here. So, preguntas.
Thank you very much for this talk. And one question is, the salt plants, the higher amount of suberin is related with a higher capacity to fix CO2?
No one has shown that relationship. But yeah, I think you have to think about it that way. I don't see how you get more roots, right?
And the other question is, suberin is composed of different phenolic compounds.
Yeah.
And my question is, this accumulation of suberin is due of higher increase of one particular phenol, or all are increased?
What's the question? I didn't get the question.
The composition of the suberin in the salt plant is different than the control plants or not?
Okay, so we think the crop plants make different suberins. You're right. Not all suberin is created equal. Okay, so things like seagrass have different suberin. So seagrass is trying to keep all that ocean water out, right? So it has suberin that is much longer with fatty acids. And so seagrass also makes 7% of their root into suberin, I think. So it's going to be a great source of carbon sequestration. So I think corn makes different suberin from pork, etc. But it all gets called suberin. Yeah.
Yes, John, thank you very much for your talk, and we are honored really to have you. I'm here.
Where? Oh, sorry.
Yes.
Okay.
We are really honored to have you here, and your lines of research you develop really impressive. And my question is related to the strategy to increase the root biomass. So you say that, of course, we expect because of greater root biomass, we expect higher carbon sequestration. And of course, you analyze the balance between sequestration and release of CO2. And my question is particularly, what is this balance? Let's say for 1 gram of carbon fixed in roots, how much CO2 in grams, let's say, would be released by respiration?
We don't know that yet. The data are not out there. Not really. Yeah. So that's a big unknown that we got a lot of criticism for in our project. But we just think, maybe if it was a better time, we had more time to work on this, then we would try to get that data before we do the experiments that we're doing now. But we don't have the time, so we have to go ahead and do them. But in the meantime, we're trying to get soil samples from all over the world, And so soil changes a lot. So it's going to depend on what the soil chemistry is, how much CO2 you can actually draw down.
Okay.
So we have to figure that out. We need to get a lot of data.
Yeah. Thank you very much.
Yeah.
Any other questions here? Yeah.
Thank you very much for your very nice talk. Very impressive. I have a question regarding the stomatal control, because when you're saying that the plant is taking the more CO2 to make more roots, what about the stomatal control? The stomata will close when there is high CO2. So that is auto-regulated when you have more suberin or more carbon into the roots?
Again, we don't have the data on that yet. So I'm trying to think. So your question is, if you make more roots, do you bring in more carbon?
Yeah, no, my question is, what happened with the regulation of a stomata? The stomata closes with more CO2.
Yeah, okay. I think that Julian Schroeder should be able to answer that question in another half a year. He found a mechanism by which CO2 gets taken up. It's a CO2 sensor. Right? So he says he has it anyway.
Yeah.
And he's looking for, what the changes are going to be in terms of water relations. Because that's going to be the way you get the answer to the stomata problem. Is that right?
Right.
Yeah, okay. So I'll have to stay on his case. I don't know. Yeah.
Okay, thank you.
Yes, thank you very much for your talk. It was really very, very interesting. As maybe you remember, they introduced us, and I work with legumes and the Rhizobia symbiosis. So you mentioned that you will analyze, let's say, other plant besides Arabidopsis. You will analyze Lotus japonicus. That is a legume and can establish symbiosis with Rhizobia.
Yep.
So my question is, do you anticipate, or perhaps another way to ask it is, will you be interested in analyzing if some of the modifications in the different traits, root traits, would modify the ability to establish the symbiosis with Rhizobia?
Yes, I'd be interested in that. Yeah, we should talk. Okay.
Well, I will read the question from the chat. Joanne, I'm here, but I read the question from the chat from YouTube.
Okay.
It's by Gustavo Rodriguez, and he asks, carbon fixation is highly dependent on gas exchange. Is the ideal plant more water consuming compared to non-engineered plant? And also, is carbon fixation in the ideal plant impaired in warmer temperatures?
Okay. You guys are asking me to solve all plant biology. I don't know. All right, so I think the temperature is going to have an effect for sure. Yeah, so I don't know what it is yet though. But plants go into stress mode when you have too high temperature or too low a temperature, right? And so that's the ethylene pathway involved in that. So I think we're going to have to keep our eyes on that. Now that we have plants, we can put them in different environments. And so hopefully that will help us figure some of this stuff out. Because some plants are going to perform better in a stress environment. That's what I think anyway. And some plants will perform slightly better in multiple stress environments. So do you want a generalist, or do you want something that's specific to some crop, that you need to get the best possible yield on? And I think the answers to those questions are different, right? So, yeah. So you still have to figure that out. Yeah.
I have a question here. It's not a science question. It's more like a maybe political science question. What would be your strategy for making this global, like technology transfer, and like convincing the decision makers, the agricultural sector to adopt and embrace this technology? Because I anticipate it's not an easy task.
Okay, so that's a tough question. I mean, we have a lot of discussion about that. It's definitely part of our program and a part that we're not trained to do, right? And so, I mean, I think that's going to be an issue. But Sequester is supposed to do that for us, at least some of that. So Sequester needs to go out there and establish relationships with big ag and small ag and NGOs, and governments. So we hope that they can do that. Because, Biden had, like, in his budget, he had a big plan for climate change. It wasn't as big as he thought, because if you look at NIH's budget, climate change is getting almost no money. And it's not going to matter if you get cancer in 5 years, because by then you're going to be dead of pollution and other problems, I mean, so I think the way you have to get the grassroots behind this. And I think that's why the activists have been getting a lot of press, but I don't like the way they go at it. I have they get mad at me because they say I'm letting people think that they can pollute the earth and I'm going to just clean it up all the time. I don't think that's my message. My message is more, we polluted the earth, so now we better clean it up, or we're not going to have an earth to be on, right? Not that you can keep on polluting it. We have to learn a few lessons from what we've done. So I think I say I'm very successful with garden clubs. So I go and talk to garden clubs all the time. Like the little town garden clubs. Like the La Jolla Garden Club or the Del Mar Garden Club. and they're very interested in what we're doing. So these are, mothers and grandmothers mostly who are in these gardening clubs, and they know a lot more about plants than I do. So I think they can be a big help. We need to have people in Washington who are doing nothing but advocating for this kind of research. And so, we have a few people whose voice gets heard, but it's hard to get your voice heard when you're just a scientist in some lab somewhere, right? I can't call up, Dianne Feinstein or whoever our senator is these days and get her to take my call, You have to have people who can get into the staff rooms of the senators and the Congress people and stuff, because it's their staff people who are going to make it happen. If somebody takes up the cause, they can make it happen. And these are often people, 25 years old. So I don't know the best answer to that, but we hired a couple of lawyers, and now we passed all them on to sequester, because we think we're good at science, so we should just do science. So yeah.
Thank you.
Well, if there's no more questions, thank you very much, Joanne, for the talk. And there should be something prepared in the lobby. I don't know if it's really there or not, but there should be something to drink and something to eat. And so you're all invited to, before you go to lunch,